Good
Good
afternoon, sir.
afternoon to all students. Yes, Adhyasya Dubey, Smarnika Patra, good afternoon. Good afternoon to you all. Okay, students, are you hearing to me?
Yes, sir.
Okay, that have you done well in examination yesterday? That means how much are you expecting from that examination? I have already checked that of 50% of your PDF, okay, and found that only 3 students that have crossed 30 plus, of which 35 out of 35 is the highest mark, and 2 students have scored 31, 32 like this, okay. So that and other students have done well, except that 1, 2 students who have not reached even the 15 marks. That means so easy questions, but why I did not have do well in that examination, that is also a question mark. Okay, I was expecting that he must
Sir,
have
who scored 35? Sir,
30
who
minutes.
scored
I'll have to tell.
35?
Yes, today we'll have to know about the results. By the evening, you will have to get the result in this that is bio WhatsApp groups. You'll have to get the results of all the students who have appeared the examination. And one student, that Abhigya Arpit Pradhan, he sent his PDF at 11 p.m. It is not acceptable. So that Abhigya Arpit, are you present here? Not present. Okay. That he sent PDF at 11 p.m. Imagine that how that he is so obedient to the teacher. Teacher gave in concession of that of 15 minutes. Within 15 minutes, that you must have to send your PDF. But that boy sent at 11 p.m. That is nearing to that of 4 hours. After the 4 hours of that of penultimate times fixed by the teacher, he sent it late. Okay, students, that so much that is a very easy question. You might have seen all the answer from your that is from the notes, and they must have corrected it. And little bit some students have made mistakes, but most of the students have answered correctly. Okay, thank you very much for that of scoring more than 25. Most of the students have scored more than 25. Okay. So students, you see that that in the last way before the
Sir,
examination.
sir, please tell now
Yes.
who scored 35?
Yes. Any problems, have you?
Sir, who has scored 35?
What you are telling, that you just write down and just show me. I'm not that understanding what you are telling. Okay, you see that. I'm just coming to my that is come to that of subject. Okay, whatever you have done, you forget it. Anamika sir, who scored 35. The student today evening you will know that who has scored 35 and who has scored how much. Okay, you wait for 4 to 5 hours, then you'll have to get your full answer. All the students who have appeared the examination will have to get their result. Don't be hurried, and that means only one student has scored 35 out of 35, and other students, 2 students nearing cross the 30 marks, and a few students have scored that is only that 50% answer sheet PDF I have checked, and I will have to check rest of it after the class, and I will have to show the result in the evening time. So don't be panic. Okay, you see that. Now come to the course. Now we'll have to discuss about that photosynthesis, that the chapter now which is going on. And you see that in the last period, that means the last class, we had discussion about that pigments involved in photosynthesis. I have told you that 4 main that is pigments, that is chlorophyll A, chlorophyll B, xanthophylls, and carotenoids are involved in the process of photosynthesis. Okay, of which chlorophyll A is that of major pigment. It is the primary pigment, whereas other 3, that are carotenoids, chlorophyll B, and xanthophylls, they are accessory pigments. Okay, their main function is to that of provide higher wavelength as well as energy to that of reaction center. Reaction center is occupied by one pigment, that is chlorophyll A. One molecule of chlorophyll A occupies that of pigment of reaction center, and that pigment is involved in that process of absorption as well as that of transformation. Absorption in the sense that it will have to observe the wavelength of light, and it will also transform that light energy into that of chemical forms. Okay, so variant energy will have to converted into that of ATP as well as that of NADPH. Okay, you know that NADPH, that is nicotine amide adenine dinucleotide phosphate. Okay, NADPH, and that will have to be happened in that of that is of a reaction center. So reaction center is attached with that of enzymes. Enzymes are present in that of reaction center, so they are also converting water into that of molecular oxygen. Molecular oxygens, and you are also getting that of electrons. So that is happened in that of that is photolysis of water. Photolysis of water is carried out in that of that is reaction center. That reaction center is also of two types. One is that of photosystem 1 and that of photosystem 2. And you see that on that that means on that day, the last period, I had also told about you that of that of different pigments. I have told you that chlorophyll A. Chlorophyll A can activate and can have to absorb more energy, more wavelength of light in that of blue light as well as that of red light. Okay, red light. And chlorophyll A is more consistent in of capturing more wavelength of light in that of red light than that of blue light. But chlorophyll A can absorb more wavelength of light, more wavelength of red light comparatively to that of chlorophyll B. But chlorophyll B is consistent of capturing and observing, that is wavelength, the blue wavelength of light, that is than that of chlorophyll A. Okay, so if you have to show in graph, you can have to understand it better. Okay, now you have to show in that of graph. You see that I'm just drawing a graph. You see that mindfully. You see that this is the x-axis and this is the y-axis. X-axis and that of y-axis. Okay, that is x-axis and that is of y-axis. And in x-axis, that what you will have to see that wavelength of light in nanometer. That is wavelength of light in nanometers. Wavelength of light in nanometers, and where you have to find out that of light observed. Okay, here is that of light observed. That is light observed. So x-axis denotes to that of wavelength of light in nanometer, whereas y-axis that is light observed. Okay, you see that. Suppose you will have to, that is 400. This is 500 nanometer. This is 600 nanometer. This is 700 nanometer. Like this, you see that. That means if you will have to show it that of that is absorption, light absorption, we'll have to mark it. You see that you mark it that of absorption of light. Okay, you see that you mark it that of absorption of light. You see that. Okay, that is the absorption of light. You see that this is that of that is blue light. Here you mark that. Here is that of blue light. Here is that of red light. So absorption. So you see that what you are marking here, that of absorption of light. Okay, absorption of light. Here you see that. Here is the in between the 400 to 500. You can mark that of blue light, and in between 620 to 720 you can mark it that of red light. You can have to that absorption of more wavelength of light in nanometer, showing to that of this is that of blue light, that is blue, and this one is that of red. And if that of absorption is more in blue light and in red, then photosynthesis rate also will be, you see that. Suppose this is the photosynthesis rate. You'll have to say that photosynthesis rate will also increase. Okay, So the rate of photosynthesis, more absorption of light in that of blue wavelength. In blue wavelength, that is, you see that maximum absorption of light. So photosynthesis rate is also higher. In that of red light also absorption, that is wavelength. You see that is in the red light. In the red light, that is wavelength of red light. Absorption is more, so photosynthesis is higher. In other cases, you mark that absorption rate is lower in that of yellow light or that of other light. You can see that absorption rate is lower, so that photosynthesis rate is also lower. Okay, so that photosynthesis rate can be higher in that of blue light and red light. And you see that is the blue light. In blue light, it is very important, students. In blue light, chlorophyll B observes, okay, observes in blue light. Chlorophyll A observes more wavelength, more light, observes more light compared to that of chlorophyll A. Okay, chlorophyll A. But in red light, chlorophyll A observes more light compared to that of chlorophyll B. Okay, that you have to remember. In blue light, that chlorophyll B observes more light compared to that of chlorophyll A. But in red light, chlorophyll A observes more light compared to that of chlorophyll B. That you have to remember. This is important. In chlorophyll B, that is monopoly of chlorophyll B comparative to that of chlorophyll A. But in red light, chlorophyll B is that of more, that is chlorophyll B is the most successful pigment in absorbing light comparative to that of chlorophyll B. Okay, so more absorption rate is a more photosynthetic rate. That is more absorption rate in blue light, so there is a more photosynthesis in blue light. More absorption rate in red light, so photosynthesis rate also higher in that of red light. That is concept you must have to remember, students. Okay. Okay, on that day I had also told you about that of students, that is of a light reaction. I had told about that of two type of reaction. One is that of light reaction and another one is that of a dark reaction. Okay, light reaction, another one is that of a dark reaction. That light reaction, students, that light reaction is otherwise known as, that is photochemical phase. Okay, that is called photochemical phase. It is otherwise known as, that is photochemical phase. Okay, photochemical phase. In light reaction, that means include what? Light reaction include what? Light reaction first into that of all, that is light absorption. That is light absorption. Okay, that is light absorptions. You see that is light absorptions. The first one is that of light absorption, then that of water splitting. Then another one is that of water splitting. That means light reaction include to that of light absorptions and that of water splitting, and then oxygen release. Another one is that of oxygen release. That is molecular oxygen, like this molecular oxygen. Okay, that is oxygen release, and then that the formation of a high energy chemical intermediate, and the formation of high chemical. Okay, formation of high chemical intermediates. High chemical intermediates like ATP and NADPH. Okay, that is That is the process of light reaction. In light reaction, it is a sequence. First, light will be absorbed. Different wavelengths of light will be absorbed. Then of water, that is the splitting of water will be split. Then oxygen release, then oxygen will be released. Okay, oxygen will be released. Then formation of a high chemical intermediates like ATP and NADPH. Okay, that is important, that you have to remember. That is, first one is that of light of different wavelengths to be absorbed by the chlorophyll. Okay, by that of pigments. Which pigments are present. Then water splitting. That means water will be split into that of that is you can have to get that of hydrogen ions, electrons, and that of oxygen. Okay, so hydrogen, oxygen, and that of electrons you can have to get it. That is water splitting. Then after water splitting, oxygen will be released. I have told you that the source of oxygen in that photosynthesis process is that of water. I have already told you that it is the most important concept you must have to understand. And then formation of high chemical intermediates. What are these high chemical intermediates? They are ATP and NADPH. Okay, this is the process. This is the sequence of that of light reaction. In a light reaction, there are four sequences. First one is that students, light will be
Absorbed.
first sequence is
Light will be absorbed.
light absorption. Second one is that of after that
Water split.
Water splitting.
yes. Then then another one is that
Oxygen release.
Release.
Oxygen release.
okay. Then next one is that
Formation of
Formation
chemical
of high-
intermediate like ATP and NADP.
yes, and that is formation of high chemical intermediates like ATP and NADPH. Okay, that we will have to understand. These are the four process in that of a light reaction. Again, in students, that it is very important that in the light system you will have to see that of photosystems. In that of light reaction, there is a most important one is that of photosystem. There are two photosystems you can have to find in that of a light reaction. In that of light absorption unit is called that of photosystem. That is photosystem 1 and photosystem 2. Okay, photosystem 1 was first discovered, so it is called for PS1. And later on that of PS2 was discovered. So in case of that of discovery, in the period of discovery, in the sequence of discovery, they have been named so. That is PS1 and PS2. That you have to remember. Okay. So that photochemical. Now we'll have to know about that of light absorption. Then we'll have to know water splitting and oxygen release. Then how ATP and NADPH are formed. Okay. You will have to know in a sequence wise. So first you will have to know that of photosystems. It is very important events in light reaction. In light reaction, photosystems are found, students. Photosystems. It is very important. Photosystems are light absorbing unit. What are photosystems? You are now going to discuss about that of photosystems. Okay, photosystems. Very important, students. Photosystems. What are photosystems? Photosystems are light absorbing unit. Light absorbing unit. Okay, photosystems are light absorbing unit, that you will have to remember. Then large complex of proteins. Then large complex of proteins and pigments. Okay, large complex of proteins and pigments. And you know that pigments are chlorophyll A, then chlorophyll B, carotenoids, and xanthophylls. Okay, these are the pigments. And we will have to later on, you will have to know about that of proteins. Which proteins that binding all that of different pigments together. Okay, that protein binds different protein together in that of photosystems. And there are two types. So light. So what are photosystems? Suppose what are photosystems? Anyone asks you, how can you have to answer? Photosystems are light absorbing units, and photosystems are large complex of proteins and pigments. Okay, large complex of proteins and pigments. That is chlorophyll A, chlorophyll B, carotenoids, and xanthophylls. You know that one molecule of that of carotene, beta carotene. Okay, carotene can give us, that is, two molecules of vitamin A. That is beta. One molecule of beta carotene can give two molecule of vitamin A. Okay, so that is carotene. So they get the four pigments. So now we'll have to know that photosystems. Now we'll have to know the division of the photosystems, about that of photosystems. So photosystem, there are two type of photosynthetic units. There are two type of units of photosynthesis. Photosynthesis are of two units in light reaction. What are they? They are photosystems are of two types. That is that is called P700, and another one is P68. P700, P68. Okay, that is, there are two type of photosystem. Photosystem 1 and photosystem 2. This one is called photosystem 1, and this one is called photosystem 2. So one is photosystem 1, another one is called photosystem 2. So photosystems are light absorbing units, are large complex of proteins and pigments. They are of two types. Photosynthetic units in light reactions are of two types. One is PS1 and PS2. In PS1, that is, reaction center is P700. In PS2, reaction center is P68. The first one was discovered, that is P700. P700 was discovered. P700, students, means 700 means that is the wavelength of light. That means this center PS1 can observe the maximum to that is 700 wavelength of light, and then PS2 systems can observe to that of 618 nanometer of wavelength of light. Okay, so into that region, as it was first discovered, that is called PS1, that is P700. As it was later it was discovered, this is called PS2, that is P68. 768 that you are seeing, that these are nothing but these are the wavelengths of light. These are the wavelengths of light, the students you have to remember. Okay, now you have known that photosystem. What are photosystems? Photosystems are
So light absorbing unit.
light absorbing units. Photosystems are large complex of dash and dash.
Protein and pigment.
Protein and pigment.
Which pigments are involved in that of photosystems? Which pigments are involved?
Chlorophyll. Chlorophyll.
Chlorophyll A,
Chlorophyll A, chlorophyll B,
chlorophyll B,
carotenoid and xanthophyll.
carotenoids and xanthophyll. One molecule of beta carotene can give how much molecules of vitamin A?
2 molecules.
2 molecules. Very good. Can you tell me that is PS1? Okay, that PS1. Are you hearing to me, students?
Yes, sir.
Okay, that PS1, you see that PS1, that is P dash, that means how much?
P700.
P700.
What is 700? That is the
Length of wavelength.
wavelength of light. That means PS1 can observe maximum 700 wavelength of light. Okay, and the PS2 can observe maximum 680 nanometer wavelength of light. Okay. Now we'll have to know about that classification of photosystems. You see that mindfully, you see that of photosystems. It is very important of the light reaction. Now you are going of that of a light reaction. That you see that is photosystem. Photosystems is made up of two components. You see that photosystems. Photosystem students is made up of two units, two components. Photosystem is made up of two components. What are these two components? First, you will have to know about that of a reaction center. First one is that of a reaction center. Okay, reaction center. And the second one is that of a light harvesting complex. A light harvesting complex. Very mindfully, watchfully, you see that. Okay, that is photosystem consists of two components. What are they? One is reaction center. Another one is light harvesting complex. Okay, and that reaction center is made up of one molecule of chlorophyll A. That reaction center is made up of one molecule of chlorophyll A. Chlorophyll A. Very important. One molecule of chlorophyll A. Reaction center has only one pigment. That pigment is chlorophyll A. Okay, chlorophyll A. But light harvest one molecule of chlorophyll A. And what is the function of the reaction center? That it will absorb different wavelengths of light and convert into that of different chemical intermediates. You see that reaction center, what reaction center capture? That is absorb light energy, okay, light energy, and a variant of energy, that is variant of energy, that it is absorbing variant of energy. And what it does, it is converting into that of chemical energy. Okay, that is converting into that of chemical energy, as well as that of chemical intermediates. What I have told you, chemical intermediates, that is ATP and NADPH. Okay, NADPH. So that transformation of light energy to chemical energy is called quantum conversion, and this type of is called quantum conversion. Quantum conversion. What is quantum conversion? That is change of light energy into chemical energy is called quantum conversion. It is very important. Okay. And that is happened in that of reaction center. What is reaction center? Reaction center is made up of one molecule of chlorophyll A, and that reaction center functions in transforming light energy into that of chemical energy and producing different chemical intermediates. Which are these chemical intermediates? These are ATP and NADPH. NADPH stands for nicotine amide, okay, adenine dinucleotide phosphate. NADPH. H means that is the reduced form of NADP. NADP reduced form is NADPH, where hydrogen ions have been added. Okay, 2 hydrogen ions have been added. So NADPH plus H plus. Okay, that is 2 hydrogen ions have been added. That is the reduced form of NADP to NADPH. Okay, that is 2 chemical intermediaries that form during that of light reaction. In light reaction, what you get? We get ATP, NADPH, and release of oxygen. These are the products of that of light reaction, which are utilized. So oxygen is released to that of molecular oxygen is released to that of atmosphere. So ATP and NADPH with that of support of carbon dioxide, that is sugar synthesis happen in that of a light reaction. Okay, light reaction. ATP synthesis in dark reaction. ATP synthesis is happened in that of a light reaction, where sugar synthesis or carbohydrate synthesis is happened in that of a dark reaction. So you know that. So that is called quantum conversion. What do you mean by students quantum conversion?
The transforming of light energy into chemical energy to form chemical intermediates is called as
Yes.
quantum.
Yes, that is conversion of light energy into that of chemical energy or intermediaries with the that is in reaction center. Okay, in reaction center is called that of a quantum conversion. You must have to write down. Okay, you should have to keep a rough note. Okay, you should have to write down what I am also writing. And that you see that reaction center has enzymes. Okay, reaction center enzymes are present in reaction center. So that is you know that is photo that is water photo that is photolysis of water, that is photolysis of water. That means water is being disintegrated into that of hydrogen, into that of oxygen, into that of electrons. So this is called what? That is photolysis. Photolysis of water takes place in that of reaction center, and it is an enzymatic process. Enzymes are involved in such process of photolysis of water, which one takes place in that of reaction center. Okay, reaction center. Only reaction center is occupied by that of one molecule of chlorophyll A. Then students will have to know about that of light harvesting complex. Light harvesting complex consists of two parts. That is light harvesting pigments. Light A stands for harvesting pigments. And here proteins. Okay, that is proteins. And the light harvesting pigments. Which pigments are present here, students? That is chlorophyll A, chlorophyll B, then carotenoids, and xanthophylls. Okay, and xanthophylls. You remember that. You will be surprised that sir has told that one molecule of chlorophyll is present in the reaction center. Why sir has brought chlorophyll A in that of a light harvesting pigment? Student, yes. You see that. That means chlorophyll A of the different wavelengths. So which chlorophyll has wavelength of 700 or 680 nanometer, they can be kept in that of a reaction center. And that is the highest wavelength. Highest wavelength in that of photosystems. In that photosystem, suppose anyone asks you that, what would be the highest wavelength in PS1? How can you be answer that one? What is
700.
the
700.
yes, 700. What is the highest amount of wavelength found in photosystem 2?
680.
680.
680. So that is the highest amount. After that, no wavelength is found. No highest amount of wavelength is found in that photosystems. Okay. That photosystem's maximum wavelength is that of 700. That photosystem's maximum wavelength is that of 680. So that this is called, one is called photosystem 1, another one is called photosystems 2. Okay, and that you see that here is chlorophyll A, here is also chlorophyll A, but you see that those chlorophyll A present in that of light harvesting pigments, their only function is to observe, not to make any chemical reaction. Okay, they cannot have any role in that of that is reaction. Okay, cannot reaction. They only have that of absorption of light. They can only do the function of absorption of light. Those chlorophyll A pigments in light harvesting pigments, they can only observe different wavelengths of light. But here those chlorophyll A present, that means it can observe different wavelengths of light, as well also can do that of chemical reaction. but this chlorophyll A can do chemical reaction. That is the basic difference in between chlorophyll A present in light harvesting pigment and that of chlorophyll present in reaction center. This is most important, students. You must have to remember. Okay, must have to remember that chlorophyll A present in reaction center is capable of absorption of light as well as doing chemical reaction. But chlorophyll present in light harvesting complex are only involved in absorption of light. This is most important point you must have to grasp. Okay, and you see that chlorophyll A, that is carotenoids and
Sir, please repeat that point.
they are called accessory pigment. This is the primary pigment. Chlorophyll A here is primary pigment. Primary pigments are called those pigments which can involve in the process of absorption and also chemical reaction. But the accessory pigments only supply the higher wavelength to that of a reaction center and help in the process of photosynthesis. They can do the process of photosynthesis. They can't carry out the process of photosynthesis. The process of photosynthesis can be carried out by reaction center having chlorophyll A. Okay, and but the chlorophyll A, chlorophyll B, carotenoid and xanthophylls, those are present in light harvesting complex. They are involved in the process of observing different wavelengths of light. That is the basic things you must have to remember, students. Okay, and you see that, and there are also protein. Okay, there are different types of proteins that are involved in such process. They are called PSA. Okay, they are called, you see that, PSAA, PSAB, PSBA, PS small b, capital B. Okay, so which proteins are what are the need and necessity of proteins? Proteins can bind all the pigments together. Okay, all the pigments can be bound together. So all the pigments can be bound together by means of proteins. Proteins can have that function of binding and keeping all that pigments intact. And which are the proteins involved in the process of light harvesting complex? That is PS small a capital A, PS small a capital B, PS small b capital A, PS small b capital B. That is photosystem 1, students. These PS A capital A, PS small a capital B, that are involved in photosystem 1. PS small b capital A, PS small b capital B, students, these are involved in photosystem 2. Okay, these four proteins are involved in that of these four proteins are involved in the process of photosynthesis in that of photosystem of light harvesting complex of photosystem. And what are these? These proteins are PS small a capital A, PS small a capital B. You can easily remember. Okay, and PS small b, here you see that instead of small a, that is small b here. Okay, A, B constant, PS also constant. Can you tell me that? Have you remembered which proteins are involved in photosystem 1?
Sir, PS small a capital A
Yes.
and
Yes,
PS small
a.
b capital A,
Ah,
then PS small a capital B,
yes.
then PS small b capital B.
Yes. You can have to tell these two proteins are a photosystem 1. PS small a capital A, PS small a capital B. These two proteins are involved in photosystem 1. In photosystem B, also that of two proteins are involved. PS small b capital A, PS small b capital B. That you have to remember. Okay. These are the proteins and that of light pigments. And you see that, students, that the photosystems 1 and photosystems 2. And you see that the number of that is in higher plants, number of light pigments, number of that is number of light pigments in higher plants. how many light pigments are found in higher plants? That is 200 to 300. 200 to 300 light higher pigments are found in light harvesting pigments are found in higher plants. In plants, 200 to 300 light harvesting pigments are found. But in bacteria, students, that is only 20 to 30. Okay, 20 to 30. In that of bacteria, that is 20 to 30. That is in bacteria. That is bacteria. Okay, that is how many light pigments are found in plants? 200 to 300. How many light pigments are found in bacteria? 20 to 30. Okay, 20 to 30. And that chlorophyll, you see that chlorophyll A, that means chlorophyll A, that is giving higher wavelength of light to reaction center. For that of chemical process is called inductive resonance. Is called, students, inductive resonance. Is called inductive resonance. What is inductive resonance? That you see that higher wavelengths given by chlorophyll A of light harvesting complex to chlorophyll A of reaction center is called inductive resonance. What is inductive resonance? That you have to tell like this, that chlorophyll A of light harvesting complex giving higher wavelengths of light to reaction center of chlorophyll A for chemical reactions is called inductive resonance. Okay, inductive resonance. That means chlorophyll A of light harvesting complex gives higher wavelengths of light to reaction center occupied by chlorophyll A for chemical reaction is called inductive resonance. Okay, it's called inductive resonance. You have to remember what is inductive resonance, students? Yes, anyone?
The
Not
chlorophyll
understood.
A of light harvesting complex giving high wavelength light to the reaction center where chlorophyll A
Yes.
has the um, work of chemical reaction is called as inductive resonance.
Inductive resonance. I am telling you again that chlorophyll A of light harvesting complex giving higher wavelength of light to reaction center occupied by chlorophyll A for chemical reaction is called inductive resonance. Okay, is called inductive resonance. So how many that is light harvesting pigments are found in plants? 200 to 300. How many light harvesting pigments are found in bacteria?
20 to 30.
20 to 30.
20 to 30. Okay, 20 to 30. Again, you'll have to know about that of a function of, and you see that two type of photosystems function simultaneously. Okay, two type of photosystems operate simultaneously due to, you see that two systems, that is chlorophyll, that is photosystem 1 and photosystem 2, that is function simultaneously. Function simultaneously due to the effect of immersions enhancements of energy. Okay, due to that of immersions enhancement of effect and red drop. I'm just telling you that it is very important, students. You have to remember photosystem 1 and photosystem 2. Okay, operate simultaneously. You write down PS1, PS1, and PS2. PS2 operate simultaneously due to the immersion, due to the enhancement immersion enhancement effect, enhancement effect and the red drop, enhancement effect and a red drop. Okay, that is very important. You have to remember. That is immersion with the scientist. That means it has made a test that the two systems to have to operate simultaneously, because one system will have to provide wavelength of light, higher wavelength of light to other systems to make these systems operative. If two systems will not operate, then another systems can provide light, that is higher wavelength of light, to other systems. So it is very important that PS1 and PS2 operate simultaneously due to the immersion enhancement effect and a red drop. Okay, so two systems operate simultaneously, not one system after another. If systems two operate simultaneously, system one will operate. Okay, that is simultaneously it will operate. If anyone will operate first, it will be followed instantly by the other. Instantly by the other, so that process can be continued. It is very important point. Students must have to be remembered. Okay, PS1 and PS2 operate simultaneously due to immersion enhancement effect and a red drop. Now students will have to know about that of functions. Okay, functions of that of photosystem. Photosystem function. Functions of photosystems. Functions of photosystems. That is functions of photosystems. Photosystems. That is point one. Function of photosystem is, students, is that light absorptions. That is light absorptions. Okay, that is important. Light absorption. Then second one is energy transfer. Okay, energy transfer. And the third one is, that is electron. Okay, that is electrons excitation. Electrons excitation. Excitation. Okay, that is three function of photosystem: light absorption, energy transfer, electron excitation. This one first, this one will be second, this one will be third. Okay, so energy will be transfer, electron, this is right. Light will be absorbed, then energy will be transferred, light energy will be transferred into chemical energy, so electron will be excited. Okay, energy will be excited. So what is the sequence of that of photosystems? First one is light absorption. Second one,
Energy transfer.
yes, that is energy transfer. Third one is that of electron excitation. Okay, light absorption, energy transfer, and electron excitation. These are the function of photosystem. Functions of photosystem: light absorption, energy transfer, and that is electron excitation. Now we'll have to know about that of students. Difference in between that of photosystem 1 and that of photosystem 2. Okay, it is very important. You have to mark it. Photosystem 1. Photosystem 1 to that of photosystem 2. Okay, photosystem 1 to that of photosystem 2. First point, reaction center is activated at that reaction center, that is reaction center is activated at P700. Okay, and the reaction center is activated at P
680.
yes, that is P680. Okay, 680. That is reaction center is activated. And then non-appressed region of thylakoids. PS1 is found at non-appressed region of thylakoids. I'm just showing you non-appressed and appressed. It is appressed. Okay, appressed region of thylakoids. I'm just showing students which are that. You see that. Suppose this is that of Suppose this one is that of okay, this one is that of mesophyll cells. This one is that of mesophyll cells. Mesophyll cells. Yes, that is mesophyll cells. Here you see that these are that of that is thylakoids. These are that of thylakoids. And this thylakoid is that of what students it is called? That is thylakoids. That group of thylakoids are called as granum.
Grana.
Granum. Okay, how many thylakoids are present? How many thylakoids are present in one granum? That is 22. That is 20 to 50. Okay, one granum consists of 20 to 50 numbers of thylakoid. Okay, 20 to 50 numbers of thylakoid. This one is called that of this one is that called of granum. Okay, that is granum. Granum. And you see that this one is called granum, and this one is, you see that, and here is another one, that is another one, this one is another one of that of thylakoid. This is also another granum. You see that one granum is joined by another granum by that of stroma lamellae. Okay, stroma lamellae. This one is called that of stroma lamellae. And this is that of thylakoid. You see that this is the thylakoid, this is the lumen, that is the lumen. The inner membrane is that of the inner side of that of thylakoid is called lumen. And after and you see that this one are the oppressed part. This one are the oppressed part. Okay, oppressed part. If one thylakoid joins with the another thylakoid, the joining part are called oppressed. These are called oppressed. Okay, oppressed. These are called oppressed. These are oppressed. These are oppressed. These are oppressed. 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 These are the oppressed. Which are non-oppressed students? Non-oppressed. Which are non-oppressed? These one. You see that? These are non-oppressed because these are free. Because no thylakoids on the down. Okay, no thylakoids attached to it. These are the non-oppressed part. These are the non-oppressed part. Non-oppressed. Non-oppressed. And this one also students non-oppressed. Okay, this one also non-oppressed. That is thylakoid. That is thylakoid stroma lamellae. That is stroma lamellae as well as stroma lamellae have also non-oppressed region. And also thylakoid. That is grana also. In the granum also there are non-oppressed region because no thylakoids are attached with it. Here thylakoids are attached, so these are that of oppressed region. That is oppressed region. So photosystem 1 is found here. So PS1. Photosystem is photosystem is found in PS1. That is non-oppressed region. Photosystem 1 is found in non-oppressed region. In that of it is found on that of outer membrane of that of photosystem 1 and also that of stroma lamellae. Okay, that photosystem 1 is found in that of thylakoid, that is outer membrane of thylakoid, as well as that is stroma lamellae. This one is called stroma lamellae. Okay, that is stroma lamellae. In that part, you can have to find photosystem 1. But here, oppressed region, here photosystem 2 are found. Photosystem 2 are found in that oppressed region. Okay, this is very important. Then PS1 is found in non-oppressed region of thylakoid. PS2 is found in oppressed region of thylakoid. Okay, oppressed region of thylakoid. And it is found in stroma lamellae. It is found in PS1 is found in stroma lamellae. Okay, stroma lamellae as well as thylakoid membranes. Okay, thylakoid membranes. But here you can have to find out that PS2 is found only in that of thylakoid. Okay, only in that of thylakoid. Only in that of thylakoid membrane. Thylakoid membrane. Okay. That is PS1 is found in stromal lamellae as well as thylakoid membrane, but PS2 is found only in thylakoid membrane. Okay, thylakoid membrane. And another point, students, you have to remember that chlorophyll A is more in that of chlorophyll A in PS1. Chlorophyll, the ratio of chlorophyll A to that of chlorophyll B. Okay, chlorophyll B is equal to, that is 2:4. That is 1:2. That is equal to 1:2. Okay, chlorophyll A ratio of chlorophyll A to that of chlorophyll B is 1:2, where it is, where in PS2 it is 2:1. So more chlorophyll. So you see that the chlorophyll content in that of 2:1. Okay, so more chlorophyll are found in that of more chlorophyll are found in that of photosystem 1 comparative to that of chlorophyll B. Here in photosystem 1, chlorophyll B are mostly found. Okay, mostly found. That then chlorophyll to carotenoids. Then ratio of chlorophyll. Chlorophyll means chlorophyll A and chlorophyll B. Chlorophyll to that of carotenoids. Okay, carotenoids is equal to 25:1. 25:1. But here is that of 5:1. 5:1. Okay, 5:1. So you have to remember that chlorophyll A to chlorophyll B ratio in photosystem 1 is 1:2. The chlorophyll ratio, chlorophyll A to chlorophyll B ratio in photosystem 2 is 2:1. That means more chlorophyll A is found in photosystem 2 than in that of chlorophyll B. Then chlorophyll to carotenoid. Chlorophyll means chlorophyll A and chlorophyll B to that of carotenoids. Chlorophyll is maximum 25:1. The ratio in photosystem 2 in between chlorophyll and carotenoids is 5:1. Is 5:1. Okay, it's 5:1. That, students, you have to remember. It is very important. How many thylakoids are present in 1 granum? That is 20 to
20 to 50.
How many grana are present in a chloroplast? That is 40 to 100. That is 40 to 100. How many grana are present in a chloroplast, student?
40 to
40 to
100.
100.
How many thylakoids are present in 1 granum? That is
20 to 50.
20 to 50.
yes, 20 to 50. Very important, you have to remember. Okay. And another point is that, students, that in the core of PS1 is associated with protein. I am just writing the core of PS1. The core of PS1. I am just writing the core of PS1. The core of PS1 is associated with protein. Is associated with proteins. Which protein, students? I have told it. How do you remember? Which proteins are involved?
So PS small a capital A.
PS A capital A. Then PS A
Capital B.
Here will be
PS small b capital A.
capital A. Then PS small b.
Capital B.
Yes. Okay. The core of PS1 is also, you see that here is a what? This is a reaction center. This is a reaction center. And it is being occupied. That means it is being occupied. That means it is encircled. All the photopigments, that is all the pigments are, that is encircled it. Encircled it. All pigments encircled it. Here is the reaction center. This one is the reaction center. Reaction center. It is occupied by chlorophyll A. And here you can have two different wavelengths of pigments. These pigments may be chlorophyll A, chlorophyll B, carotenoid, or xanthophylls. You see that. That means the light. See that light. That means this one will give to that of highest pigment. This one will give like this. This one give like this. this one give like this one give like this, or this one give this one, this one give this one, this one give this one, this one give. Then they will give light to that of reaction center. Okay, you see that here is that of that is wavelengths of light. That light, some rays, that means higher wavelengths. The smaller one given to that higher one. This higher one given to that higher one. This higher one given to that. This one give to that reaction center. Ultimately, all are giving their wavelengths of light energy to that of a reaction center. So it is encircled. That means reaction center is in the middle and all are surrounding it. That you know that this is the light harvesting complex. That is light harvesting complex. The main function of the light harvesting complex is that of to provide energy in the higher wavelengths, but to safeguard the reaction center from photooxidation. It is very important. Light harvesting complex most important function is that to keep it from photooxidation. Okay, safeguard from photooxidation. Okay, photooxidation. You see that reaction center is in the middle. All are, that means all that of a light complex, that is light harvesting complex or the pigment surrounding it. I have shown you in one side, but it is covered in all sides. Reaction center is middle. From the all sides, that higher wavelengths of light being given by that of pigments to reaction center. So they also keep the reaction center from that of photooxidation. Okay, that is from photooxidation. That is very important, students. You must have to, okay, you have to safeguard from photooxidation. That is safeguard from photooxidation. Photooxidation means suppose more intensity of light comes to that of a reaction center. So reaction center will be damaged. It can tolerate up to that of a 700 or 680 nanometer wavelength. Suppose 800 nanometers wavelength light come to that reaction center, so it will be damaged. It will be broken down. So no photosynthesis will take place. So such pigments surrounding the reaction center, they will protect us. Okay, they are the bodyguards. Act like bodyguards to that of reaction center. You have to imagine in such concept. Okay, that of photooxidation, safeguarding or protecting reaction center from photooxidation. Photooxidation means if more intensity of light, high intensity of light comes to the reaction center, there is every chance of damaging the reaction center and stopping the photosynthetic process. So in such way that such light harvesting complex, such pigments will have to be ready to protect the reaction center. That is the main motto of that that is light harvesting complex. So in case of Photosystems 1 and Photosystem 2, the core protein, that is the core protein, the core of PS1 is associated with proteins. You have to remember like this way. Okay, this is the very important one, students. You must have to remember. Okay, and the energy needed for the excitement PS1 is more than PS2. Okay, that is this is very important point. The energy needed for excitement, the energy needed. It is another important point. Energy needed for excitement of PS2 is more than that of PS1. Okay, energy needed for excitement of PS2 PS2 is more than PS1 because shorter wavelength of light because shorter wavelength of light needs more energy. Okay, wavelength of light requires more energy. Okay, light wavelength of light needs more energy. More energy. It is very important, students, that concept you must have to be remembered. Energy needed for excitement of PS2 is more than that of PS1, because you see that is PS2 is shorter wavelength 680, so it will have to take more energy. Short wavelength, more energy. More wavelength, less energy. Okay, less energy. In such concept, you must have to understand. That is, energy needed for excitement of PS2 is more than that of PS1, because shorter wavelength of light needs more energy or higher energy, high energy, more energy, whatever. So PS1 needs less energy. So reaction center of PS1 needs less energy. Reaction center of PS2 needs more energy. Higher wavelength, less energy. That less wavelength, that is lower wavelength, maximum energy. That concept should be clear. This is, students, all about that of photosystems. Okay, photosystems. In the next period, tomorrow, we'll have to finish that of that is photosystems 2. We can have to find out that of photophosphorylation, that is cyclic photophosphorylation, non-photo that is phosphorylation. You'll have to know about that of that is photolysis of water. You can know about that of chemi-osmotic process in which ATP is synthesized. Tomorrow you can have to know. Very important concepts are coming tomorrow. So you should don't take this chapter lightly. You should have to go through whatever I am teaching on that day. Same day you should have to go to through the notes and to that of what you are writing, what you are understanding. You should have to make up in your mind. Also rethink about it. Then you can have to build up your concept. Okay. So that only 1-2 questions you have to do today, and we will have to finish the class. Okay. Now ready for that of questions. Okay, students, come to that of question number 1. Okay, that is question number 1. Select the first question about that of select the correct pair. Okay, that is select the correct pair. Select the correct pair. Okay, I'm just telling you, just answer. Prophase equatorial plate. That is prophase equatorial plate. Okay, equatorial plate. Then you see that metaphase, appearance of nuclear envelope. Metaphase, appearance of nuclear membrane. Nuclear membranes. Okay, metaphase. Then come to anaphase. Anaphase. Separation of sister chromatids. Sister chromatids. Okay. Then option D is telophase. Okay. Telophase. Telophase. That is breaking down of, okay, that is breaking down of a nuclear envelope. That is nuclear envelope. Okay, students, can you give the answer? Right answer, anyone? Yes, Adhyasa Dubey, she. Then Smaranika Patra, she, right answer. Then anyone? Anyone? Okay, that means no one. That is Mona Lisa Rautra, she. Adhyasa Dubey, the brilliant answer. Beula Khora also right answer. Abhinas Barik also right. Sanbhi also right. Okay, that right answer you have given, but the first answer given by that of Adhyasa Dubey, brilliant answer. Adhyasa Prophase 1. In Prophase 1, that is breaking down of nuclear membrane. So Prophase 1. So Prophase 1 is wrong. That is equatorial plate is in that of metaphase. Okay. Appearance of nuclear membrane found in telophase. So anaphase, separation of sister chromatid. So answer C is the right answer. Okay. Answer C is the right answer. Then come to the next question. Which of the following is not a significance of mitosis? Next question, students. Which of the following? Next questions. Which of the following is not a significance of mitosis? Okay, of mitosis. Which of the following is not a significance? It produced new cell for the healing of wounds. Okay. A. It produces new cells for healing of wounds. Okay. Then option B, it brings about variation. It brings about variation. Okay. Then option C, students, it helps in maintaining the size of the cell. It helps in maintaining the size of cell. Okay. Option D is that it replaces the old. It replaces the old and worn out cells. Option right answer, students. Which one is the right answer? Yes, Sanbi D, no. Smaranika Patra, no. Adhyasa Dubey is the right answer. Brilliant answer, Adhyasa. Mona Lisa, wrong answer. Okay, that is Adhyasa Dubey. That Beula Khora also right answer. Okay, that you see that significance. It produces new cells for healing wounds. It is true. Okay, it is true. It is the function of mitosis. Again, student, you see that it brings about variation. Mutation, variation is that of is related to that of meiosis. Genetic variation is related to that of meiosis only. Okay, in mitosis, all are looking alike, all are of equal. But in meiosis, due to that of division of sex chromosomes, segregation of sex chromosomes, there is a variation. Variation is only found in meiosis, students. Okay, so this one is the right answer. This one right, because this is for meiosis. This statement is for meiosis. That is not mitosis. All are. That is helps maintaining the size of cell. That is true for mitosis. Replaces of old worn out cell. This is also true about mitosis. Which one is wrong for that of mitosis, students? That is brings about variation. Variation is brought about. Genetic variation is brought about by meiosis, not by that of mitosis. Okay. So brilliant answer given today by Adhyasa Dubey. Okay. She is very quick in answering all the question. Many thanks to him. All the students also who have given the answer, that right answer, many thanks to them also. Those students who have failed to give the right answer, also I am giving you thankful for their participation. Okay, and so know the varieties of question, concept of question. Understanding the concept of question is very important, rather than how much or how many you are correcting here. Okay, thank you very much, students, for joining. Thank you.
Thank you, sir.
Thank you, sir.
Thank you, sir.
Thank you. Thank you.